Alright guys, let's see how this goes. Today we're on chapter 16, which is about the evolution of populations. So at this point we've talked a little bit about how evolution works. Now we're going to talk about how populations change over time. So first off, there are two main types of evolution that we're going to be discussing today, and we're going to kind of come back to this a little bit. We're going to talk about macroevolution, and we're going to talk about microevolution. Now, macroevolution is basically where you get a new species, so it's change above a species level. So yeah, it's where you go from to having a common ancestor and having that ancestor split into an entirely different species. So you could say these guys have a common ancestor and they have been split into two species. And then there's microevolution, which is honestly much more common, and that is any change in allele frequency, and that's going to be within a species. So it's like when one color comes more dominant than another color, or not more dominant, more prevalent, you could say. And then maybe that'll switch back like it did with the moths, where the other one became more prevalent after the switch back. So yeah, those are our two main types of evolution. Now, here's an important distinction when it comes to evolution. Individuals are selected, but it's populations that evolve. So let's kind of keep in mind that individuals are never going to evolve. If a bear needs a fur coat, it doesn't mean that a bear is going to get a fur coat. Maybe a mutation in the population, but then that population would have to spread. And just kind of keep in mind why this is. Mutation is a slow process, not only because you have to wait for random mutations, but maybe that mutation is a recessive mutation, right? And then you have to have like two to actually have the trait. And then so the only one that would exhibit any difference in fitness would be this one. And so it takes a lot of time sometimes for these mutations to really take a hold of a population, especially if there's not necessarily a whole lot of or especially if it's a recessive trait. Okay, so what we're going to talk about today is really going to put together what we learned about genetics and what we've learned about evolution so far. So one of the things that Darwin really struggled with when he was coming up with his theory, and one of the things that we've really had to supplement in Darwin's theory, is how traits were passed from one generation to the next. Darwin didn't understand, because the understanding really wasn't there yet, how traits got passed on. So what we could do is we can combine Mendel's ideas with Darwin's theory of natural selection, and that really completes the picture of evolution quite a bit. So evolution, especially in this case, since we're going to really talk a lot of microevolution today, evolution is any change in allele frequency. And so what we really have to do is determine, if we want to know if a population has changed or evolved, is determine whether the frequency has changed or evolved. And so the question becomes, can we calculate that frequency? Of course we can. And we're going to do that with the Hardy-Weinberg equations. So just hold tight, maybe put this in your notes, because Hardy-Weinberg equations are coming. So Hardy-Weinberg equations are used to calculate allele frequency to determine if evolution is occurring. So I mean, and that's what we'll be doing, is we'll be seeing, did they evolve? Did these change over time? Okay, so let's move forward. So here is a bunch of alleles in a population. These are diploid organisms, you could tell, because they have one copy of each gene. So let's go ahead and sweep. Okay, so P is going to be the frequency of the dominant allele. So you're going to want to write this for sure. So P is frequency of the dominant allele. So in this case, it would be the frequency of the big R's. Q is going to be the frequency of the recessive allele. And when we see frequency, what we're really talking about is how common. So how common is the dominant allele, and how common is the recessive allele? So if you counted them all up, there are 12 big R's out of 20 total R's. So it has a frequency of 0.6. That's how frequent it is in the population. And the Q, there are 8 little r's out of 20 r's, and so it has a frequency of 0.4. And one of our first Hardy-Weinberg equations, and honestly the easiest one, and the one you're going to use the most, doesn't that feel good? This is the easiest one and the one you're going to use the most, is P plus Q equals 1. And you could see that 0.6 plus 0.4 equals 1. And that makes sense since these are all just fractions. I mean, it all adds up to 20, so it should be 1 at the end, right? So since those are just fractions. Okay, so can we figure out genotypes, though? How many big R's there are? Or how many little R's? Or how many big R, little R's? How do we figure out, or how do we get those diploids? Remember, is through fertilization. The sperm entering the egg, each one bringing an R with them, right? So this is all the possible male gametes. Males could have a big R or a little r. Now, if you take that and you multiply it by your female chances, where you could get a big R or a little r, you're going to get this equation. So this is actually the second Hardy-Weinberg equation. So you can go ahead and write that down. It's P squared plus 2PQ plus Q squared equals 1. And then we can talk about what each of these parts are. For sure you need that. So P squared is the frequency of homozygous dominant individuals. So in this case, it would be the frequency, or how common is big R, big R. 2PQ is the frequency of the heterozygote individuals. So that would be how many big R, little r's there are. And then Q squared would be the frequency of the homozygous recessive individuals. So they would be the little r, little r. So you're really just figuring out how common are these guys, how common are these guys, and how common are these guys. And remember, it's going to add up to 1 because that's 100% of the population. There's no other options. They're either big R, or big R, little r, or little r, little r. So that's going to add up to everybody. So that's your second Hardy-Weinberg equation. And those are really the equations we're going to worry about. So our two important equations are P plus Q equals 1 and P squared plus 2PQ plus Q squared equals 1. So just kind of to keep in mind when we're going to use these equations. If we're wondering about the frequency of alleles, you're going to use this equation more often. And this is really what we'll be looking at a lot too. It helps out. This equation helps out a ton. Or there's the frequency of the genotypes, the diploid version, which is this equation. So just kind of like keep that in mind. So we're going to calculate this stuff. So how many flowers will be white, little r, with just this information? So remember, this is the frequency of the big R, and this is the frequency of the little r. And P plus Q equals 1, I can tell there. So when we're doing this, I'm always going to suggest we make a little chart off to the side. If we can solve these things, this becomes a really easy problem. So this is my little Hardy-Weinberg cheat chart, all right? And this is all funk. I don't know who else does this, but this is just how I like to organize this information. So Q, we know, is 0.4. P equals 0.6. And so we can use this little thing off to the side to help us figure out, okay, well, what's little r, little r? So little r, that was the frequency of the homozygous recessive genotype. And so that was Q squared. So that's really what we're looking for here. How many flowers will be white? And we want how many. Now, I don't know. That's a really interesting way of putting that question because I don't have a number of individuals, but that's kind of common. Well, let's figure out, instead of that, let's figure out the frequency of the white flowers because that's something we can actually figure out. Okay. So really all we have to do then to figure out little r is just square that, right? So 0.4 times 0.4 is just going to be equal to 0.16. You could do it on a calculator if you really need to, but that's 16 and 2 decimals, so it's 0.16, okay? And I mean, that answers the question. You could figure out the rest, but that answers the question. So if I say, okay, in a population of 300 flowers, how many flowers would be white? Well, all you got to do is go 300 times 0.16, and that would tell you how many flowers would be white. So that's a pretty powerful equation. Look at that. Oh, look, how many flowers would be red? Oh, well, why are you doing this to me? Let me go back so I can show you what exactly they were doing. So they went and figured out the red too. I thought that's rude. So yeah, let's figure out red while we're at it. Well, okay, so red would just be p squared, so big R, but don't forget that there's two ways to be red, right? Because you could be red to be big R, or you could be red to be big R, little r. So actually, red flowers are going to be p squared plus 2pq. That's going to tell us how many red there are. So actually, we have to add that all together. So anyway, to figure out p squared, well, you just square that. So 0.6 times 0.6 is just 0.36. 2pq is just 2 times p times q, so 0.4 times 0.6 is 0.24, times 2 is 0.48. All right, and so to figure out the total number of red flowers, you just add these guys together. 14. 4 plus 4 is 8. And that's 0.84. So 84% will be red flowers and 16% will be white flowers. Another thing we could have done is if these are all the white flowers, we could have just done 1 minus 0.16, and that also would have given us how many red flowers. I always overcomplicate, but there you go. Okay, so some Hardy-Weinberg tips, some things you're going to want to be careful about. You'll want to make sure to decide if they're asking about percentages or individuals. So they're asking for frequencies, which is a decimal, 0.16. Are they asking for percentages, where that would be 16%? Or are they asking for individuals, where I wonder, out of 400 individuals, how many will be this? And then you have to take that 0.16 and multiply by that 400 to figure out how many individuals. Okay, so you've got to be careful about what they're asking for. Like here, 1 in 1,700 Caucasian newborns have cystic fibrosis. Big C is the normal allele, dominant over the recessive little c. Individuals must be homozygous recessive allele to have the disease. So it could ask what percent of the above population has cystic fibrosis. It could ask how many individuals have cystic fibrosis. Or they may ask what is the frequency of the recessive allele. And if that's the case, all they're asking for is Q. Or the frequency of the dominant allele, where they're asking for P. So there's all kinds of things that they could ask for. So you'll want to kind of make sure what they're looking for. So make it a point to find these 4 things first, and you'll have a really easy time. Like I said, make this little chart off to the side, and you're going to have it really simple. And another thing, make sure you always find the recessive information first. And kind of let's remember, why would we want to find the recessive information first? It's because we already know, if I'm saying that it has the recessive phenotype, what the genotype must be. Because if I say red is dominant to white, right? Red could be big R, little r, or big R, but white, you can only be little r. So if I say, all right, 200 of them are red and 150 are white, well, out of these red, you don't know who's big R, and who's big R, little r. So it's really hard to figure out the P stuff first. But white, you know that all of those guys have to be little r. So it's actually really easy to figure that guy out, because you know anybody that's white is little r. So always go with the recessive one first. So let's do a few practice problems. In a population that is in Hardy-Weinberg equilibrium, so that's going to come up. If it says Hardy-Weinberg equilibrium, that means it's not evolving, okay? And if it's not evolving, that means that the frequencies are going to stay the same. That means they're not going to change. So let's look at this. The frequency of the homozygous recessive genotype of a certain trait is 0.09. So that's Q squared is 0.09. Okay. What is the percentage of individuals that are homozygous for the dominant allele? So let's kind of keep in mind we're going to have a percentage here, and we're looking for the dominant allele. So that allele dominates the P's. So P squared is the genotype, big R. The allele is P. So what we're looking for here is P. That's what we're going for. And we're looking for a percent. All right, so from here, it's pretty simple to solve this problem. to figure out Q, you just have to take the square root of that, and that would be 0.3. You could do that on your calculator if you really need to, but trust me, 0.3 times 0.3 is 0.09. Okay, well, if P plus Q equals 1, then 0.3 equals 1 P. So we just have to solve for that, and P equals 0.7. Okay, and then define P squared is just going to be 0.7 times 0.7, which is 0.49. Okay, and then we could do 2 times P times Q. 0.3 times 0.7 is 0.21 times 2 is 0.42. And now we figured out this chart, we can go ahead and answer the question. So homozygous dominant or percentage of homozygous dominant, so that is 0.7 for the frequency. Oh my gosh, you guys are letting fun go crazy. It's not the dominant allele, it's the homozygous. So this is big R. Wait, so actually it's this that we're looking for. I lied. So that's 0.49, but that's not a percent. That's not a percent. That's a frequency. So that's going to be times 149%. Okay, so let's try again. Hopefully Funk won't get confused this time. So I'm always going to start with my little chart off to the side because, man, does it help to create this little chart off to the side. Okay, allele for big W for white is dominant over little w for black. In a sample of 900 sheep, 891 are white and 9 are black. Whew. Estimate the allelic frequencies. So that's P and Q. So I want this in this sample, assuming that the population is in equilibrium, meaning it's not going to evolve, so these numbers are going to stay the same. So here we go. To figure out Q squared first, because we're always going to start with Q squared first, because it's easier. So Q squared are all going to be the homozygous recessives, so that's all going to be these guys. So Q squared is going to be 9 sheep. Now, how do I figure out what the frequency of that is? How do I take that from numbers of individuals to decimal? Well, okay, it's just 9 out of 900, so you just divide that, and that'll tell you how many are Q squared. This is me being totally lazy because I don't want to find it. So I'm going to grab my calculator here. 9 divided by 900, because laziness. 0.01. Oh, I could have figured that out, but I didn't want to. Okay, so Q squared equals 0.01. And then from there, you can just keep going, because take the square root of that to figure out Q, and you got 0.1. Well, P plus Q equals 1, so that means P must be 0.9, because 0.1 equals 1, P, right? Okay. That means that P squared is just going to be 0.9 times 0.9. So 9 times 9. Shoot. I am so tired. Is that 81 or 89? Oh my gosh, I'm a horrible teacher. Shh. Don't tell your math teacher. Thank you. It's 81. I was like, is it 81 or 89? Okay. So that means that this is going to be 0.81. And then 2 times P times Q is just going to be 0.9 times 0.1 times 2. So that's 0.09 times 2. So that should be 0.18. Yeah, so that's 0.18. Okay, and so that is that. Now let's answer the questions. So we want the allelic frequencies. Oh, well, we already did that. It's 0.1 and 0.9. See? So simple, so easy to solve. Okay, so what factors lead to a change in allele frequency? Like, what causes evolution? And let's kind of remember that equilibrium is when a population is not evolving. So if these numbers never change, then we would say the population doesn't evolve. So let's say that Q squared is like 0.12, and then 100 years later, Q squared is still 0.12. Well, then we say, okay, over 100 years, that population did not evolve. But if it starts out 0.12 and 100 years later, it was 0.24, well, then we can say, oh, there was a shift in those frequencies, and so that population changed over time or evolved. Now, There are certain factors that would lead to evolution, and there's certain factors that lead to non-evolution. Sorry, I have to stretch. Very tired. So, these are how you get equilibrium, the rules for equilibrium, the rules for no evolution. All five of these things must be true if you want the population to not be evolving. So to not evolve, the population has to be a large population size. Small populations tend to evolve quicker than large populations. There needs to be no gene flow, so no organisms moving into the population and no organisms moving out of the population. You can't bring extra genes in or there'll be evolution, and you can't take the genes out or there'll be evolution. No mutations ever, because mutations give selective advantages. There has to be random mating always. Somebody can't have an advantage when it comes to mating, or there will be selective advantages. And there needs to be no natural selection. Everybody has to do just as good as everybody else. So, if all five of these things are true, then we can expect that the population will not be evolving. If 0.12 is the frequency of the recessive allele, and then if this is all true, 1,000 years from now, it should still be that. All right, so a population to not evolve, these things have to be true. And so a question we should ask ourselves, well, is this very likely? And probably not, especially because most of this is not within any, no ecosystems have no mutations, okay? So this is a really idealized event and something that you're not likely to get. So let's talk about how these things do this. So if you have a small population size, you're subject to something called genetic drift. So everybody please say genetic drift, which is random changes in the gene pool or random changes in those allele frequencies. And it's just due to a small size. Like there's a random volcano and that just randomly happens to kill all the brown bunnies instead of all the white bunnies, okay? And so that was just a random thing. The volcano didn't care what color the bunny was. It just randomly happened to take out those. And if there had been a lot more bunnies, well, then maybe it wouldn't have just randomly happened to take out all of the white. I forgot what color got taken out. Okay. So let's talk about what a bottleneck is really quick. So sometimes small population sizes leads to a bottleneck, and that's where a small population ends up with a smaller gene pool. So the original population might have had quite a few variations, quite a few different genes and alleles, but there's a bottlenecking event which kills off most of the population. Most of the population dies off, and so only a few survive. And of the few surviving, you lose a few genes. You lose a little bit of genetic variation. And we actually saw this happening with the cheetahs. They were hunted almost to extinction. It had an original genetic diversity. They were quite a diverse group, but then they got hunted quite a bit, and we lost a lot of the alleles. And so they're actually pretty similar now. And they're much more subject to evolution, and they're much more subject to things going wrong, too. So that's a bottleneck. Then there's the founder effect, okay? The founder effect is where the newly founded populations don't always represent the genetic diversity of their sources. So it'll say like this population gets blown away in the wind over to an island, and it just so happens that only the red beetles got blown over to the island. Well, these founding members of this new population will all be red, and this new population won't have any of those green beetles. So the founder effect can lead to a change in frequency because you've gone from maybe about 50% red and 50% green to 100% red. So this new population changed or evolved. Okay, you also have to have random mating, so you can't like have organisms choosing which organisms they want. So that's called sexual selection, by the way. Sexual selection is where there are adaptive changes that lead to increased ability to secure a mate. And that's obviously going to cause evolution because if you're choosing which male is more attractive to mate with, you're going to make much more attractive males. Okay, so it's called female choice, where they choose the more attractive males. Sometimes it's the other way around. Keep in mind that sometimes our beautiful colors and stuff can be our downfall. Like peacocks, they have to get away from tigers in the wild. but their tails make it really hard to get away from tigers. The tigers get a lot of peacocks that way from their tails. So too much of a good thing sometimes could also send evolution the other way and make towards smaller tails maybe. All right, so keep in mind there has to be no natural selection. Nobody can do better than anybody else if you want no evolution. So keep in mind natural selection is a mechanism that leads to adaptations in our environments where some traits are more fit and so those traits become more popular. So how could selective pressures lead to different results? Well, think of all the different possibilities that could lead to light mice in this population, or all the different pressures that could lead to dark mice, or maybe pressures that lead to the middle colored mice. So that's what we're going to talk about really quick is this types of selection. So I would just have a quick types of selection page there. Just a second. Just want to check something. Oh, come on. Come on. There we go. I was checking how close we are to being done because I know I'm tired. We're close. Okay. So the types of selection which we're going to talk about today are directional selection, and you can see that disruptive selection and stabilizing selection. So directional selection, you can see it's where we favor one end of the spectrum. So in this case, the darker mice were favored and all the light mice died off. So that is natural selection moving the population in a certain direction. So natural selection favors one extreme of the phenotype. All right, then there's disruptive selection, and that's where natural selection favors both extremes. And so the light mice do really well, the dark mice do really well. So that eventually there's going to be a lot more dark mice and light mice and a lot fewer of the middle ones. And this, by the way, is a lot of the times how we get new species, because these guys start becoming so different from each other over a lot of time that eventually they wouldn't be able to mate with each other. And then finally, there's stabilizing selection. That's where natural selection favors the middle. Okay, so the light mice all die off, the dark mice all die off, so you have a lot more of the middle color. Okay, now what preserves genetic variation? How do we get a lot of variation? Because variation is really important to evolution and survival. Well, to preserve genetic variation, one of the greatest things that has caused a lot is diploidy, the fact that we have sexual reproduction, two parents, mixing the DNA from those two parents. We end up with independent assortment, all kinds of stuff. And we're also going to talk about balanced polymorphism, which leads to something like heterozygote advantage. So let's talk about diploidy really quick. Diploidy is where the is one of the things that really creates a lot of variation, because unfavorable recessive alleles can hide in the heterozygote. So even though this is an unfavorable trait, that trait gets preserved because it still just hangs around in the heterozygote. So it's hard to get rid of traits if they're not too awful, or if in the heterozygote those organisms are just fine. So it's actually a rare event if this gets expressed, and so it keeps this frequency really, really low. Especially if this is harmful, then that frequency is going to stay really, really low. And then last slide, guys, balanced polymorphism. Okay, that's basically where we maintain stable frequencies of different populations, because hey, sometimes there is an advantage in being a heterozygote. Because remember, sickle cell anemia, especially in places where there's malaria, it's not necessarily a good thing to have normal red blood cells because you're susceptible to malaria. It's definitely not a good thing to have only sickle cells. It's actually an advantage to have both. And so that's called balanced polymorphism, and it's what preserves certain genes in a population, even though they might be bad in certain respects. Whew, we made it. Good job, guys. That was nice and long. So make sure you go back if you need any help, and let me know if you need anything. Thank you.